This lecture is an introduction into cellular membranes and how substances are able to transport in and out of them. So one of the main questions I want you to ponder while going through this lecture is, why are cells so small? Looking at the scale, proportion, and quantity of different objects again, you can see that eukaryotic cells land between 10 and 100 micrometers. But why not 1 millimeter? Why not 10 millimeters? Why not 1 meter like a human? Why are cells restricted to this size window? Something to think about. Another question I want you to ponder: when we think of the great diversity of cells we find in life, we find a tremendous variety of shapes and sizes. Look at the neuron in the thyroid cell compared to the skin cell. Why is this the case? Why so many different structures? So the cell membrane is going to provide us some insight into these two questions: why cells are small and why cells come in so many different shapes and sizes. The cell membrane, also known as the plasma membrane, is the barrier between the inside of the cell and the outside of the cell, and it's going to be able to regulate movement. Specifically, a cell has certain substances that need to move in. It needs to have food, such as sugars, proteins, fats, and salts, oxygen and water as well for all those chemical reactions, and it needs to move substances out. It needs to get rid of waste like ammonia, salts, carbon dioxide, water, and any products it produces, such as proteins, to ship to other cells or other vessels in the body. Big picture here: cell needs that membrane to regulate what materials go in and what products or waste go out. But how is the cell going to do this? How do you build a membrane that's going to be able to keep the watery contents of the inside of the cell separate from the watery contents out? Because remember, cells are mostly water, and so too is the aqueous or water environment they're surrounded with. What kind of a substance is going to be able to do that, to not immediately mix with water? Think back to the molecules you've already learned. Well, lipids, fats, they do not mix with water because they're nonpolar. Because of this natural phenomena of nonpolar lipids, lipids are going to be the molecule that forms the foundation of the cell membrane. It's going to be a specific kind of lipid called a phospholipid, and you can see two different diagrams of them here. You can see the simplistic one on the left and the more accurate one on the right. A phospholipid is made up of two primary parts. The first part is it has a phosphate in its head region. Sometimes it's signified by a circle in the sea jelly-like model. And you can find it at the top of a more accurate molecular rendering. That phosphate head is polar, and that makes it hydrophilic or water-loving. That's the portion of the membrane that will be attracted to water. Attached to that phosphate head are fatty acid tails. And these fatty acid tails are nonpolar. They are just chains of hydrocarbons. Since they're nonpolar, they are hydrophobic or repelled by water. They're not going to mix it. Because of this, the phospholipid is what we call amphetic. It has two regions, each that have a different property. The top, or the head, is attracted to water. It's polar, and the tails are repulsed and nonpolar. These in a cell membrane are arranged in a bilayer, or a layer of two phospholipids. Notice how the heads are pointing towards the water and the tails point towards each other. This is again because of those natural properties. The phosphate is polar, so it's attracted to the polar aqueous solution outside. Same thing here with the inside, but the nonpolar tails will stick together. This maintains the integrity of the membrane. You have a nonpolar region that is not going to mix with water and remain stable. To give you a more zoomed in look at this phospholipid bilayer, we have those two polar hydrophilic regions pointing outside and in, and the central non-polar region where polar activity is not going to occur. This is what gives us a cellular barrier or border that's going to be able to keep inside in, outside out, and regulate what is entered by chemical means. But since the membrane is built of fats, just like you saw, lipids can freely flow through that membrane. Other objects, though, like sugar, water, salt, and waste, are not going to be able to easily, if at all, cross that impermeable nonpolar barrier. Polar things cannot mix with nonpolar. So this is going to be able to regulate and define what enters and what exits the cell. Because of this, we say the cell membrane is a semi or selectively permeable membrane. What that means is that some substances can move through, but not all. The membrane is selecting what is permeable or what can move through and what cannot. Think of it like a coffee filter or a sieve. If I had put coffee in a coffee filter, run water through it, small particles are able to move through the filter, but the larger coffee grounds stay behind. Same idea with the selective or semi-permeable membrane of the cell. So the cell needs to have certain things frequently move across. So let's keep in mind what those things are. Sugars for cellular respiration need to make their way in. So do lipids, amino acids, oxygen, water, salt, and waste needs to be able to freely move out. So how can we regulate these while blocking everything else out? Well, some molecules, like you saw before, can directly move across. Fats and oils can directly move across because they are made of the same substance as the cell membrane. Here's why this is significant. Later on, we'll look at how steroids, lipid-based communication molecules, are so powerful in communicating and triggering cells to do actions. The reason why hormones are so powerful is they can move directly across your cell membrane. They don't have to wait, they don't have to attach to anything else, they can directly go in and cause a reaction. Other stuffs are typically blocked out immediately unless they're given help. Sugar, amino acids, and salts, and waste. But water is a special case. Almost all cells have special channels called aquaporins that allow water to freely move in. And even when a cell doesn't have aquaporins, research has shown that water molecules, because of how tremendously small they are, can actually push through that non-polar tail region and make it through the cell membrane, like you're seeing in this video here. It's a struggle. That molecule is moving around, but it can make it to the other side in small amounts. To get a large molecule across the cell membrane, the cell's going to have a doorway or a tube for those substances to move across. It's going to accomplish this by embedding proteins into the cell membrane to act as a tube for large molecules to move through. Like you can see in this diagram in the bottom left, these proteins form channels upon which objects can move. Here you can see the sugar molecule is now able to move across because that tube's going to prevent it from interacting with the non-polar tails in the membrane. Another unique thing about using proteins is they are specific. Remember that the three-dimensional structure of proteins, that structure determines its function. Each channel has a unique shape that can only fit one object. Think of that children's toy where you have a cylinder, you have to fit in the cylinder hole, a triangle, you have to fit in the triangle hole. Triangle's not going to fit in the cylinder hole. Same thing here with specific molecules. So there are channels that are specific to amino acids, and only they can fit through. Specific to water, those aquaporins I told you about, for salt and for waste. Another way cells are able to regulate how much stuff goes in and out is by how many of these protein channels they make. If a cell detects food, well, they're going to make more protein channels so they can get more food in. But if they don't detect any food, well, that's a waste of energy, so they'll just let those break down and have fewer channels. So the number of these protein transport channels do vary between cells and at different times in a cell's lifetime. In order for these proteins to embed into the cell membrane, they need to have the correct molecular polarity and structure. Specifically, within the membrane, this red region here, that part has to be nonpolar on the outside. The R groups of that protein is nonpolar so that it can attach to the nonpolar fatty acid tails. The inside of the protein is a completely different story though. It can be polar to facilitate the movement of polar particles. The outside of the protein similarly needs to be polar. By being polar, it's able to anchor to the polar phosphate regions of the phospholipids. So the outside of proteins mirror the cell membrane, polar on the outside, nonpolar on the inside. However, the inside of the transport protein can be polar or nonpolar based on what material it's carrying. There are two different classes of proteins. Most proteins are able to go all the way through the cell membrane, but others will only attach to the outside. Proteins that only attach to the outside of the membrane are called peripheral proteins. They're loosely bound to the surface. Their function is not to transport substances, but could be to adhere the cytoskeleton to the cell membrane or as a cellular name tag so that the cell is able to be identified by other cells. Proteins that go all the way through are called integral proteins. They integrate all the way through the cell membrane. These are your protein channels. They're often referred to also as being transmembrane proteins. This is what's going to let substances move in and out, or what's going to act like a pump. But proteins have so many different varieties and functions in the cell membrane. Here's just a few. I mentioned to you it can be a transporter. It can be a channel to get substances across the polar and nonpolar regions in the cell membrane. But it can also be a site for enzyme activity. Molecules can attach to the proteins in the cell membrane, and they can be broken or built up. It can also be used for cell communication. One end of the protein could receive a molecule, and the other end could trigger a response in the cell. They're also used as ways for cells to identify each other. We use visually looking at our faces or the sound of our voices. Cells use molecules. Proteins are also used for cells to stick to each other. If I have something like skin where cells need to pancake or stick on top of each other, proteins can be a binding site for those cells to hold together. And as I mentioned before, it can be used as an attachment for the cytoskeleton to provide structure from the inside of the cell to the inside of the cell membrane. A little more about cell recognition. Cells need to be able to identify each other. Your immune system needs to know, is this just a healthy skin cell, or is this an invader that needs to be destroyed so you don't get sick? To do that, there are carbohydrate chains that we call antigens that are attached to proteins on the cell membrane. This is how different cells are going to be able to recognize each other. Is this a friendly cell, or is this a bacteria or a virus that needs to be destroyed? They come in two different varieties. One variety is called a glycoprotein. Glycoproteins are carbohydrate antigens that are attached to a protein. Name makes sense. Glyco, sugar, protein, protein. The other variety is a glycolipid. These are antigens that attach directly to a phospholipid. Glyco, sugar, lipid, lipid. Now, the cell membrane faces a problem with temperature. Cells have to maintain homeostasis. They need to have just the right temperature for all their activities to occur. So the cell membrane needs to make sure it doesn't get too hot or too cold. Why? Well, if it gets too cold, those phospholipids can move closer together. The cell can become too rigid and might break apart. And if it becomes too hot, the phospholipids will move further apart. They'll become too flexible, won't hold their shape, and the cell will leak out. To overcome these problems, cell membranes have cholesterol in them. The cholesterol molecule acts as a fluidity buffer in the membrane. It counteracts the close proximity or too far of a distance that a cell might encounter if things get too hot or too cold. For example, if it gets really warm and things start moving apart, adding cholesterol will actually force those phospholipids to move closer so that they don't completely move apart and the cell leaks out. Same thing when it's cold. When it's cold, the cholesterol acts as a buffer, preventing those phospholipids from moving too close and the cell breaking. Yes, eating too much cholesterol is bad for your health, but a small amount of cholesterol is necessary to maintain the fluidity of your cell membrane to make sure it doesn't get too close or too far when it gets too hot or too cold. So pulling together all of these structures I mentioned to you, we represent this model of the cell membrane as being the fluid mosaic model. By fluid, we mean the membrane is flexible. All those phospholipids are moving around and the membrane can bend. By mosaic, we say that it's made of a bunch of small parts that when viewed further away, make up a larger structure. You can see this beautiful mosaic of toilet paper, one of our most sacred and scarce resources. It's made of millions, well not millions, but many, many small tiles. But when you look far away, you see the image of toilet paper. Same thing with the cell membrane. We see phospholipids, cholesterols, proteins, and when you look far away, you just see that outer membrane on the cell. So I've gone through the primary structures and their functions of the cell membrane. To check yourself for your test, on this model, could you identify and describe all of these structures? If so, you're ready for your test. If not, you might want to rewatch parts of this video. Looking immediately at it, I know this is a peripheral protein because it's attached only on the outside. This is a integral protein. It goes all the way through the cell. It's probably a transport channel. Here I can see phospholipids in a phospholipid bilayer. There are cholesterol molecules in there to maintain the fluidity of the membrane. I can see a glycoprotein used for cellular recognition. It's a sugar attached to a protein, and a glycolipid, that cellular ID tag attached directly to a lipid. And down here, what you're seeing are microfilaments from the cytoskeleton attaching to a protein to give it that structure. So now that we understand the structure of the cell membrane, we need to understand how movement occurs through it. Specifically, why do molecules move through the membrane to begin with? If I give them a channel, why would they move from the outside in? Things don't happen spontaneously or randomly in life unless there's something driving it. Specifically, there tends to be a tendency moving from particles from a high concentration to a low concentration. If there's more stuff outside the cell than there is in, the more stuff outside tends to move in. This is something we call diffusion, or the natural movement of particles from an area of high concentration where there's more stuff to an area of low concentration where there's less stuff. And you can see it in this animation. All of these yellow particles start concentrated in a high concentration in one corner and naturally diffuse or move out to where there's a lower concentration. This happens with dye as well. If I put dye in an aqueous solution, a water solution, those particles will move from a high to low concentration until there's an equal amount of stuff everywhere in a state that we call equilibrium. Why does this occur? Well, this is because of the second law of thermodynamics. The universe tends to go towards disorder, something we call entropy. This is more subject of physics and chemistry. In biology, just know this is how it occurs because it's a law of nature. Things go from order to disorder. What's unique with life, though, is we temporarily go in the opposite direction. We go from disorder to order, and then we die and return to disorder. When this diffusion occurred, was any energy required? No. This just happened because of the second law of thermodynamics. All living systems are constantly needing energy, constantly don't want to expend more energy than they can obtain. So for movement like diffusion to occur for free, this is something that living systems have evolved to take advantage of because there's a huge energy benefit in not having to spend any energy to get particles to move. This type of transport with no energy required is frequently referred to as passive transport because no energy is being expended. It just occurs passively. So what I showed you before was simple diffusion, or moving from high to low. In this scenario where I have 3 fat molecules inside the cell and many outside, what direction would the fat move according to diffusion? Well, it's going to move from high to low. So where there's more fat, it's going to move until there's an equal amount on both sides and we've hit equilibrium. And it didn't require any energy on behalf of the cell. Now, what if you're too large, though, to cross the membrane by yourself? You're going to need a protein channel to move across. For example, sugar. Sugar is going to get blocked by those phospholipids unless there's a protein channel available. But it's not going to freely move on its own. Something needs to cause it to move. If there's a protein channel available, and there's a concentration on one end that's higher than the other, those sugar molecules will move by diffusion through the transport protein. This is called facilitated diffusion. Why? Because the protein channel is facilitating or helping that diffusion to occur. That diffusion can't occur without the protein channel because the sugar can't make it across the membrane, but with that protein channel, it's being helped and can diffuse. So these are 2 different types of passive or no energy required transport. Simple diffusion is just moving from high to low across the membrane. If a protein channel is required, diffusion occurs from high to low across a protein channel, and we call it facilitated diffusion. But things don't always work this way. Sometimes you need to move in the opposite direction. Instead of moving from a high to a low concentration, you need to move from a low to a high. You need to go against entropy, against the laws of the universe. To do this is going to require some energy. So we refer to this as active transport. With active transport, a protein channel is going to act as a pump. It's going to spend a little energy in the form of ATP, or adenosine triphosphate, to push things from a low concentration to a high concentration. When would this occur in the cell? Well, cells like you are storing extra nutrients in food in case they go a while without eating. A cell is probably going to have more sugar in it than is outside. but it wants to get that sugar in. It to constantly have all the resources it needs. So it'll use active transport to push the few sugar molecules outside of it inward to store for later against its concentration gradient. A reminder from chemistry, concentration gradient just means if I have stuff, what is the concentration at any region? So could you identify and describe the three types of transport I just covered? You can see all three in this diagram here. Well, I know the top one, I see particles moving directly across the membrane from a high concentration to a low concentration. So that's an example of simple diffusion. In the middle, I can see the same thing moving from a high to low concentration, but it's occurring through a protein channel. So that's facilitated diffusion. Both of those are passive transport because no energy was expended by the cell for either. This bottom mode of transport, though, I have the movement of low concentration particles to an area of high concentration. The cell's spending ATP to do that, so I know that is active transport. So going back to the question we started with, why are cells so small? Why can't a cell be as large as a human or as small as an atom? The cell membrane has everything to do with it. Look at these three cubes here. These cubes were soaked in a chemical over the same amount of time, and wherever the outside chemical diffused, that part of the cube turned white. You can see a small cube, a medium cube, and a large cube. Here's the percent diffusion that happened for each size. The 1 cubic centimeter cube diffused about 50%, about halfway through the cell. 2 centimeter, only about 25%, and the large one only about 10. So what effect does size have on the rate of diffusion across that cell membrane? Everything. Smaller cells are able to efficiently diffuse food in and waste out. But notice that the larger cells, they weren't able to readily diffuse as much. I only had 25% diffusion in the medium and 10% in the large. But why? Well, it deals with the surface area to volume ratio. Surface area is the area that makes up the surface of the cell, and the volume is its cellular contents, the space it occupies. If you were to calculate the ratio of its surface to its volume, there's a clear pattern. Small objects have a very large surface area to volume ratio, meaning they have more surface area compared to their volume. But as you get larger, you have less surface area to your volume. Cells have to be small because of this. A cell can't be large because at a large scenario, they cannot transport materials rapidly outside them and inside them. They can't get enough substances in and enough waste out. But at a small size, they can rapidly diffuse. And this works with human body heat as well. If you have a lot of surface area but very little volume, you're probably someone that gets cold very quickly. Why? Because your surface area is constantly giving off heat, but you don't have much heat generation with your volume. But if I had a lot of volume and very little surface area, I would probably be warmer in colder environments much more rapidly. Why do cells exist in so many different shapes and sizes? Well, same idea here, surface area to volume ratio. Cells like the neuron and the thyroid, by increasing their surface area, they're maximizing the area where they can transport materials in and out, or in the case of the neuron, to rapidly send messages to other cells. The opposite principle is used as well. If you decrease your surface area, you're lowering the amount of transport, and we see this with cells that are involved with protection like skin cells, and this is partly why a lot of skin cells are just dead cells there to protect you.